Welcome back to the second presentation that is covering Blossom Chapter 3. Recall that last time we discussed sections 3.1 on the cell membrane. Spent a lot of time there, really talking about the structures of the cell membrane, helping you understand that some molecules are easily permitted to travel across the cell membrane or to leave the cell, and other molecules are not allowed to travel through the membrane, or at least it's highly regulated through different processes like diffusion, facilitated diffusion. We talked about endocytosis and exocytosis. Also, we spent time looking at the cellular organelles, those little factories, those components of the cells, each of which has an important role. And recall, your job is to recognize them and know what those different roles are. And then finally, we looked at the nucleus, and we looked at the first process that is covered in this chapter, and that is DNA replication. In addition to DNA replication, today we're also going to continue our conversation, and we're going to be looking at protein synthesis. We're also going to be looking at cell division, think mitosis, and we're finally going to be taking a look at cellular differentiation. And these topics are really important take-home lessons that are a part of this chapter 3. So let's jump into protein synthesis. This starts with section 3.4 in your Blossom. And as the name suggests, this is the process by which proteins are going to be synthesized by your cells. Recall, we have trillions of cells. Each of those cells has its own metabolism, and each cell is responsible for making its own proteins. Think of it, remember that building up things is part of your metabolism, specifically, or anabolic reactions, or your anabolism. So protein synthesis is a great example of anabolism, where the cell is producing proteins from the building blocks that were gained through biochemical reactions and through your diet, right? From the molecules that are absorbed into your body and then are broken down by your digestive system through different catabolic reactions. So, where do proteins come from? The instructions to build proteins come from the DNA. So in the nucleus of your cells, you have DNA. DNA is the blueprint for all of what your cells do. The DNA contains the instructions for everything that your cells produce. Okay? And we're going to talk about how DNA takes or provides instructions to the cell, and through this series of instructions, proteins are made. In order to make proteins, there are actually two processes involved, and I'll go through both of these. The first one is transcription. The second process is translation. So I'll spend some time and we'll break those two processes down. Now, you could spend, like replication, you could spend an entire exam covering the details of transcription and translation. I'm not going to expect a deep dive into all of the gory steps, into all the enzymes, and into all the regulation of transcription and translation. Instead, I want you to walk away with an overarching appreciation of these processes. What is happening? Why do we care? And I think by the time we finish our time here now, you will better understand why this is an important part of understanding anatomy and physiology. Why is it that we need to understand these processes? Is because when problems occur in protein synthesis, oftentimes there's a disease state, there's some problem in the body. So we need to understand how these processes work. So your DNA. We looked at DNA briefly last time. Let me remind you, going back to 3.3, DNA is that long molecule, right? It's a long, long molecule. It is composed of ACTs and Gs, these bases that make this long chain. If you could take all of the DNA in one of your cells and stretch out the DNA, it would be approximately 6 feet long. So we have to understand that that 6 feet of DNA is chopped up into 23 pairs of chromosomes, and those chromosomes are all tightly packed into the nucleus of the cell. DNA, remember, is not just the DNA molecule, but also has proteins wrapped around it. Those proteins are called the histones. And together, the DNA plus the proteins make this molecule called chromatin. So chromatin is really the component, or what you would say your chromosomes are composed of. So in your chromosomes and along that DNA are the instructions for making proteins. Along that long molecule of DNA, along the chromosome, there are a number of sections called genes. We've all heard about genes, right? Genes are little functional units. They are the building block, or they are the most important part of your chromosomes. Each gene is responsible for constructing one or more proteins. So the genes provide the instructions from the DNA to make the proteins. When a gene is turned on, when a gene is we can say the switch is flipped, when the gene is turned on, we say that the gene is being expressed. So gene expression is basically the process of taking the information on a gene and ultimately making protein. So you could say that gene expression is very similar in its meaning to protein synthesis. All of the genes found in you that make you human, you could call the human genome. So if we take all the genes are the human genome. If we take all the proteins in your cells that make you human, we would say that's the human proteome. All of the proteins in a cell would be its proteome. All of the genes in a cell would be the genome. You'll see these words a couple of times as we go through. Definitely watch this little video on what is a gene. It will help you get a better and a visual understanding as well. Now again, we're not going to go into the deep dive of this, but we need to understand that the information on the chromosome, in the DNA, in the gene, is set up in a sequence or in groups of 3 bases. We call this the triplet, okay, the triplet. So the whole basis of how DNA encodes the instructions to make proteins is based upon a 3-letter sequence called the triplet. So a triplet is simply 3 bases, ACTs and Gs. And let's take a look at this, and I'll show you some other examples of this at the end of our time today, and I think this will all make sense. It may not make sense yet, but it will as we finish up today. So here we're showing a short representation of a segment of DNA. Imagine that this is part of a gene. Remember that DNA is double-stranded, so we're seeing on the top TAGCC, right? We're seeing the sequence here, and on the bottom we see what's called the complementary sequence. In DNA, T's, thymines, always match up with adenines, A. So you'll always see T and A's matching up. Cytosines and guanines also always match up. So you'll always see C's and G's matching up. So this is the double-stranded molecule of DNA. In the cell, DNA undergoes a process called transcription. In transcription, the top strand of the DNA, in this case, is read by a series of enzymes to create another molecule called mRNA, or here it's just showing RNA. So the T matches up to be A. The A matches up to be another base called uracil. Uracil, U, is only found on RNA. Backing up, T's, thymines, are only found on DNA. So that's one very distinctive difference. You can always tell if it's an RNA molecule if it contains U's. You can always tell if it's a DNA molecule, it will contain T's, and you don't find both U's and T's on the same molecule. So from the double-stranded DNA, transcription makes a single-stranded RNA molecule. A single-stranded RNA molecule. Now, this process of transcription occurs in the nucleus. I'll show you a picture of that in a moment. From the nucleus, this single-stranded RNA molecule is going to leave the nucleus through those nuclear pores that we saw last time in the nucleus or around the nuclear envelope. And this mRNA, this RNA molecule, is going to leave the nucleus and go out into the cytoplasm. And there is where translation will occur. So we see two processes. First, transcription, followed by translation. And translation happens on the organelle, you know it as the ribosome. And recall that ribosomes are oftentimes found associated with the rough ER. So translation happens on the ribosome out in the cytoplasm, and the ribosome will then translate this single-stranded RNA sequence into amino acids. So here we see the three-letter abbreviations for amino acids. You're not responsible for learning these three-letter abbreviations. I'll show you the magic code, the genetic code that the cell uses to undergo this change from DNA to RNA to protein. So that's the big story. Let's just keep going, talk just a little bit more about a few more details. Again, DNA is found in the nucleus. DNA is double-stranded. Through the process of transcription, an intermediate molecule is made, and you can think of it as a message, right? The DNA contains all of the information, and from the DNA, a segment of the DNA called a gene sends out its message through this intermediate called mRNA. So mRNA is a single-stranded nucleic acid, and it carries the instructions that are going to be responsible for telling the cell what protein to make. And here we're just seeing a graphic of an RNA molecule. You're not responsible for recognizing this, but we see a single strand of bases, and we see the presence of uracil. So again, wherever you see uracil, you know that it's an RNA molecule. It turns out that there are a number of different kinds of RNA. I'll show you a table for this in a few minutes. And this video goes through those three main types of RNA. So how does this happen? Here is that picture once again of DNA. So DNA, remember, is a very long molecule. And here we see that double-stranded molecule. And we're just going to focus in on this little segment. So this is a gene. This is a part of a gene within the DNA. And enzymes come along, and they're going to read just one of the two strands of the DNA, and from that create the RNA transcript. So this picture represents what we saw up here. Right up here, we saw that double-stranded DNA. One strand of it was used to make the mRNA or the RNA molecule. So this schematic here is now part of it is being shown here as we take a look at the one strand of the DNA is being read along by an enzyme that then makes the RNA transcript. Now, there are some things, again, that I'm going to cross out. I am not going to ask you to know the stuff that is in blue. Okay? So I'm crossing out these steps, these specific steps. Oops, and I did it again, didn't I? I need to remember, after I hit blue, to hit save. And you need to do the same thing. If you're going to highlight something in your BLOSN, make sure that after you highlight it, that you then hit save. So what is happening here? This RNA molecule that is being made, it contains the instructions. But one thing I haven't told you yet is that the DNA in your chromosomes and the genes in your chromosomes actually has little pieces that are separated by segments of DNA that are not important. So once the RNA is made, part of the RNA needs to be cleaned up. It needs to be processed. And in this processing, we call this splicing. So we're now going to take part of that RNA molecule and we're going to remove the non-coding regions. You can think of it as the junk, right? So the non-coding regions, some books might refer to that as the junk. And what we're going to do is we're going to get rid of that junk. It's being done through a mechanism. We're not going to worry about the specifics of this, but here's a picture of what happens. Here is the RNA that was made. And the RNA is broken up into segments called exons and introns. Exons and introns. If I were naming this, I would have chose a different word. In the way that this works, the intron sections are the parts that are going to get spliced out. They are removed in this processing, in this process. The exons are the coding sections, and they are going to be maintained. So in this processing, right, the splicing process, all of the green colored introns are going to be cut out. All of the coding segments, called the exons, are going to be spliced together. So as this process finishes, we end up with the final RNA sequence. And we see that the exons are back to back, the introns have been cut out, and the cell will now use this final spliced RNA molecule to be translated to make the protein. Again, the couple pictures are coming. Let me get through this wordy section, and then we'll take a look at some slides as well. So once you have made the mRNA, that mRNA molecule is going to leave the nucleus, and it is now going to undergo the process of translation. Translation happens out at the ribosomes, and therefore you can say that translation always takes place in the cytoplasm, right? It happens out in the cytoplasm associated with the ribosomes. Most of the ribosomes are attached to the rough ER. Other ribosomes are just floating around in the cytoplasm. So this process of translation can happen either on the rough ER or it can simply happen sort of floating out in the cytoplasm. Ribosomes are little nuggets of protein and RNA. And the kind of RNA that is used to make ribosomes is called ribosomal RNA. Recall last time I told you that ribosomal RNA is composed, it's made by the nucleolus. Remember, the nucleolus is a separate organelle found inside the nucleus. So this ribosomal RNA, it's a different kind of RNA, is made by the nucleolus inside the nucleus. So far we've seen two types of RNA. We've seen this mRNA, the messenger RNA, and we've seen ribosomal RNA. There's a third type of RNA in this story as well, and it is called transfer RNA. I'll show you this in a moment with some nice images. But transfer RNA is going to carry or ferry or transfer the correct amino acid over to the ribosome. So as the protein is being knit together by the ribosome, the tRNA is going to attach, carry the correct amino acid to the ribosome. I think it's easier for me to show you this in pictures in a moment, and there are some outstanding videos here that will also help you visualize it. So make sure you do spend some time taking a look at these videos. They really will help you pull this story together. Here we're seeing a small segment of the mRNA. We know it's mRNA because it says that it is. We know it's mRNA because it has U's in it and it's single-stranded. And the ribosome is composed of two parts. There's a large part and a small part. And the mRNA literally goes through the two parts of the ribosome. As it passes through the ribosome, the process of translation happens. Here's where the magic is. In the mRNA, remember there's that three-letter triplet. That three-letter triplet is called the codon. It contains the code. This is the magic of this process. So these three letters, A, U, G, they represent a triplet, three letters. We call those three letters a codon. The tRNA, shown here in the green, is also made of RNA, and it has on one end of it a three-letter sequence, a triplet, called the anticodon. And notice that this particular anticodon is UAC. Well, UAC is the perfect complementary match to AUG. So these three letters, AUG, on the RNA are the codon. The tRNA, and there's a bunch of these different tRNAs, is going to have the anticodon, and the anticodon that has this UAC on it always carries this amino acid met to the ribosome. So we're beginning to see sort of what is this code that allows for the DNA and RNA message to bring in the correct building blocks for making proteins. So we go from the first three letters to the next three letters. UUU, that's the codon, would match up with AAA. And whenever UUU is in the RNA, it is going to bring in an amino acid called phenylalanine or Phe. And then the next one we see is CGA. And there'll be yet another tRNA that's going to bring in the appropriate amino acid to help build the protein. Each of these amino acids is then connected together through a chemical bond as the protein is made. So that is, again, a very quick overview of the process of translation. I've mentioned the key players. I will bring up those key players again in some images as we go along. And again, your job is to understand the big idea of these processes. This video talks about the steps of translation, those steps of translation being initiation, elongation, and termination. And like I did before, we're not going to worry about, I'm going to make that blue, so don't worry about these steps and don't worry about the details included in this particular video, 3.31. So let's take a look at a picture here. Let's put this all together. And this, my friends, is really the level of understanding I need you to have for these processes. We're looking at a cell. The cell has a nucleus. A very simplified image here. We have a segment of DNA. You know that there'd be a whole lot more DNA, but a small segment of DNA, this would be part of a gene. That gene is undergoing transcription to make a single-stranded molecule of mRNA. What's missing here is that this diagram does not talk about or show the processing that was included to make this final copy, but we trust that that has happened here. And notice that the transcription process occurs inside the nucleus. Once the mRNA is made, remember there are nuclear pores in the membrane of the nucleus. The mRNA is going to come through those openings. It's going to come out into the cytoplasm where it is going to find the rough ER. The rough ER is not shown here. All we're showing is a ribosome. This is the large part of the ribosome. This is the small part of the ribosome. The red molecule passing through the parts of the ribosome represent the mRNA. And as the mRNA passes through the ribosome, it is translated and the appropriate amino acids are added as the protein is synthesized. Okay, so again, two processes, transcription followed by translation. Transcription is in the nucleus, translation occurs in the cytoplasm. And that, my friends, is what I want you to understand for the big ideas of protein synthesis. I'm going to keep going, and then I'm going to recap these ideas in a series of other images that I'll share with you. There are some PowerPoint images, and I will also post these PowerPoint images. They'll be part of this presentation today, and those PowerPoint images you're going to find posted below this video in the lecture folder. Let's move on. Don't forget to answer those questions at the end. Okay. Moving on, let's go to cell growth and division. You are going to be seeing some information about cell growth in Lab A. In Lab A, you're going to be learning about the cell cycle and you're going to be learning about mitosis. Now, these are topics that all of you have had at some point in the past. It may have been a while, but these are all basic biological ideas that are important, and they're all incorporated into that Lab A. So let's talk about this. Remember that cells are they're the smallest, most fundamental living thing in your body. Your cells, each of them has its own metabolism, has its own response to environments, right? And is made up of complex structures. We've been talking about these organelles. In addition, remember that cells have a life cycle. They grow, right? Just like we have a life cycle as an organism, each of our cells has a life cycle. Now, some cells in your body, once they are formed, they live forever. Or at least they live until you die, or they live until they die off, but they're never replaced. And one example of cells that once they're formed are not replaced are your neurons, the nerve cells of your brain. So when you sustain brain damage, the brain does not have a mechanism of growing or replacing cells that have been damaged by trauma. So as we get older, we start losing neurons in our brain. But most cells in our body have a mechanism that once they're damaged, more of those cells can be made. So most cells have a growth cycle. They grow, more cells are made. Those grow and are replaced by other cells during our lifetime. Most of the cells in our body, in fact, all of the cells in our body are considered somatic cells except for the cells that produce egg and sperm. So those are called germ cells. But what we're talking about here right now are somatic cells. Soma means body. So your somatic cells are really just your regular old body cells. They're the vast majority of all of your cells. Somatic cells, all the cells in your body, have 2 copies of all of the chromosomes. We say that these chromosomes are homologous, right? Homo, they're the same. And that because each of our cells has 2 copies of these chromosomes, we say that these cells, these somatic cells, are diploid. Di meaning 2. So a diploid cell has 2 copies of all the chromosomes, right? 2 copies of the chromosomes. Each cell in your body has a life cycle. It lives for so long. Some cells in your body only survive for hours, maybe a few days. Some cells may last for 3 or 4 months. Some cells may last for a few years. but almost every cell has a cell cycle where it at some point is going to end its cell cycle. Typically, at the end of the cell cycle, the cell will make 2 more cells. Okay? And we'll look at this in a diagram and better understand this idea of cell cycle. So cell cycle occurs through the process of mitosis. Mitosis is the process of cellular division. In mitosis, one cell makes 2 identical daughter cells. So one skin cell makes 2 skin cells. One muscle cell makes 2 muscle cells. One stomach cell makes 2 stomach cells. The process is mitosis. We'll look at the stages of mitosis. That's very much a review component for this Lab A, and I'm rehashing it here. And when mitosis is completed and the cell has gone through all of the steps to make 2 identical daughter cells, there is a moment where the 2 cells divide, and that moment we call cytokinesis. Cyto, cell, kine or kinesis, movement, right? So you may know someone who studies kinesiology, that is movement of the body. So cytokinesis is the movement of cells, 2 cells becoming distinctive, separate from each other. So let's take a diagrammatic look at the cell cycle. Okay. So the cell cycle is divided up into 4 parts. M is mitosis. M is mitosis. We will break that down into further steps in a moment. Once mitosis occurs, those 2 daughter cells are going to split in that process of cytokinesis, and then each daughter cell is going to begin its own life cycle, and that begins with this G phase. G phase. G stands for growth. Some books will call it the gap phase. So the growth or gap phase. During this time, the cell is growing. It's doing what cells always do. It's doing its metabolism. It's responding to its environment. It is doing all the things that cells do. Some cells get into G1 and they do a sidebar out to what we call G0. And these are cells that will never divide. And I've already mentioned neurons. The nerve cells in your brain are like this. So nerve cells do a little exit off G1. They become G0 cells, 0 meaning they're never going to divide. But most cells stay in G1. So the cell's going to grow, and at some point, the cell is going to get big enough to think about dividing itself to make 2 more cells. But before the cell can divide, it must go into the S phase. The S phase stands for the synthesis phase. And the synthesis phase is where replication happens. Okay, so the S phase is where cellular replication happens. We know what that is, right? That's when the cell makes an exact copy of the DNA. Because, right, the cell is about to divide. And when the cell divides, it needs to have 2 complete copies, not only of the DNA, but during the synthesis phase, the cell also has to manufacture more mitochondria. It has to divide, it has to get more endoplasmic reticulum. It needs to have more lysosomes and peroxisomes. It has to have everything so that when it does divide, it can quickly divide the pie in half, and each of the new daughter cells has a complete complement of all of the necessary components for that cell to start its own life. So once the cell comes out of S phase, it has duplicated everything and specifically has replicated the DNA. The cell then goes into a second growth phase or gap phase, G2. This is not as long. And basically during the G2 phase, the cell is making the final preparations for dividing, and then it will enter into mitosis. So now this one cell will go into mitosis. Mitosis is the process of cellular division, and again, 2 daughter cells will be produced. Again, this life cycle or this cell cycle can be hours to years in length. It varies depending upon the cells in your body. So nice little conversations down here about G2 and G1. And so that's what's happening in the stages of the cell cycle. Now, mitosis. Mitosis is this time of cell division, and you're going to be looking at mitosis. And when you look under the microscope at a cell that is actively undergoing mitosis, you can see the chromosomes. This is the only time in the cell cycle where the chromosomes are visible to you under the light microscope. If you are in G1, S, or G2, the chromosomes are so loosely arranged that you're not able to see the actual chromosomes. So let's remind ourselves what chromosomes look like, right? A chromosome. A chromosome is a highly wound up, in this case, a wound up molecule. It has, remember, it has the DNA and the associated proteins in it. And here we're seeing the two copies of the chromosome. So pink and blue, one of your chromosomes comes from your mom, one of your chromosomes comes from your dad. We call those matching chromosomes homologous, right? Homo, the same. They're slightly different, but they have all the same genes on them. So we see here a pair of homologous chromosomes, right? And all of the cells have two copies of all the chromosomes in them. So we call that a diploid cell. Let's take a look at mitosis. So mitosis is a relatively quick phase. It's just a couple of hours for everything we're about to describe to occur. So let's take a look at these stages of mitosis. Prophase. I'm going to skip over prometaphase. I'm not going to worry about prometaphase at all. So I'm only going to worry about prophase, metaphase, anaphase, and telophase. These are the four stages that I'm going to care about. So we have prophase. Pro, before, right? It's preparing for mitosis. Then we have metaphase, anaphase, telophase. Telophase is the end of mitosis, and then the cell will divide into two at a moment called cytokinesis. So let's take a look at what we see here. In prophase, make this a little bit larger. In prophase, you can see that the nucleus is got a bunch of dotted lines on it. The nucleus begins to disappear, starts to break down. Okay, because remember what we need to do. In mitosis, we need to take those chromosomes that are inside the nucleus and we need to split those chromosomes into the two daughter cells. So we have to get rid of the nucleus. So one of the things that happens in prophase is that the nuclear envelope breaks down. Also, the chromosomes condense. They get tightly wound up together, and for the first time, they become visible. So if we can see the chromosomes, and we can see that the nucleus is breaking down, and the third thing that happens in prophase is that the centrosomes, these are the organelles here, they look like little bugs, they're going to go to each pole of the cell, to the north and south pole of the cell. Okay, then we go to metaphase. And in metaphase, it's a very distinctive look to metaphase. In metaphase, the chromosomes line up along the, quote, equator of the cell. So the equator, or the metaphase plate, is this plane within the cell. All the chromosomes line up, and it's very distinctive. At the same time, the centrioles that have now migrated to each end of the cell are going to send out microtubules. Those microtubules are called the mitotic spindles. And those mitotic spindles are going to reach out to the chromosome. That's metaphase. Then in anaphase, those microtubules, those spindle fibers are going to grab onto the chromosomes and pull the pair of chromosomes apart. So here, metaphase, M for middle. Here, anaphase, A for apart. The cell begins to look more like a football or a rugby ball. And the chromosomes are clearly being pulled apart toward what will become the two daughter cells. Telophase, telo is a prefix that means end or last. So telophase is the last phase of mitosis. And you can remember it that way, or you can remember T for two, because in telophase, we're about to make the two daughter cells. Notice that what happens in telophase is the opposite of what happens in prophase. In telophase, we begin to see the nucleus reform. So the nucleus, you can see dotted lines, is reforming. The DNA begins to unravel, starts to decondense, and will no longer be visible. And all this is happening, right? And then those mitotic spindles begin to break down. We don't need them. So those are going to break down or dissociate as well. And then at some moment, the cytoplasm is going to start to divide. We call this moment cleavage, right? For obvious reasons, right? It looks like cleavage. And when the cleavage is complete, we would say that cytokinesis has occurred. And now you have two daughter cells. Those two daughter cells are now independent of each other. They contain all of the organelles. They contain a complete set of DNA. Their nucleus is rebuilt, and they're about to start their own G0. So once we finish mitosis, and we now have two daughter cells starting their own G1 phase. So those cells are a little bit smaller, and they are younger, and they're going to begin their own life cycle. So those are the stages of mitosis. Everything I said is written out here, prophase, and then going down, everything about metaphase, anaphase, and finally telophase, finishing up with cytokinesis. Now, if I said it, it's important. If there are extra details here in the reading that I did not point out to you, then you're not going to be directly responsible for those things, okay? So for example, they, in the book, use the word kinetochore. I am not, didn't talk about it, so you can cross it off. It won't be something you'll need to worry about. This next section discusses the control of the cell cycle. I have already blued it out. Remember, blue means to you don't need to worry about it. I'm going to talk about this in a slightly different angle in just a moment, but you're not responsible for the material in between or the material here in blue. But I will talk about the cell cycle here in a minute, okay, when we go on and talk about cancer. So let's talk about cancer just a little bit, and I'll share with you some other slides in a few minutes. So what is cancer? Right? We know cancer affects many, many people. It takes many people's lives. What is cancer? Cancer is when a cell loses control of its cell cycle. The cell cycle is this beautiful orchestration of cells dividing and making new cells, and it's highly regulated. Cells don't just divide because they feel like it. Cells divide because there are very careful signals that the cell sends. And then cells go through their life cycle, and they undergo apoptosis, a word I'll describe to you in a minute. It's the cellular death process. And they go through apoptosis, and all of this is highly regulated. But when a cell loses control of its cell cycle and it begins to grow uncontrollably, right? Think a tumor. A tumor is a single cell in your body that has gone crazy. It's lost control of its cell cycle, and as such, it starts to divide and divide and divide and divide. So that's what cancer is, and I'm going to share some other ideas with you. But it is a problem, a disease, really a genetic disease, where the cell loses control of its cell cycle. And we probably all have horrendous family members and stories that we can tell about our family members who have suffered with cancer and the side effects of chemotherapy. So I am going to now go to 3.6, and then I'll jump to the PowerPoint slides for a few minutes. So in your body, in your bones, for example, you have cells waiting to repair your bones if your bone gets broken. In your muscle, you have cells that are waiting to replace and repair your muscle should you damage your muscle. You all know that if you cut your skin, that you heal very quickly. If you burn inside your mouth, you know that within a day or so, you've forgotten that you burned your mouth on that hot coffee, right? It heals itself. Those are those cells that are waiting and come to the rescue to repair your tissues are called the stem cells. Okay, and most of the tissues in your body, most of the organs in your body contain stem cells that are there to replace damaged tissue. Now, there's two exceptions here, or two examples that have very few stem cells. One is the brain, and one is cardiac muscle, your heart. So if your heart gets damaged, it doesn't really have the capacity to make a new heart, right? It can't really repair itself. And the same is true of your brain. Once your brain sustains damage, there is no opportunity for the brain cells to undergo mitosis. Recall, brain cells, nerve cells, go into that G0 phase, and as such, they never will divide. Even if they're damaged, there aren't stem cells there to help repair a spinal cord or brain damage. Now, these stem cells come from the early embryo. So you were first formed from the coming together of one sperm from your dad and one egg from your mom. That first cell, the first fertilized cell, is called the zygote. And the zygote is a single cell that we say is totipotent. Totipotent. Total potential, can become anything. Right? So totipotent, it has the potential to become any of the cells in your body. This is a very, very cool story. Early on, that embryo is going to divide basically into two parts. Part of that embryo is going to continue to divide to become you, and part of that early embryo is going to dedicate itself to becoming the placenta. So very early on, there's a division that occurs. Some cells will become the placenta. Some cells will continue to become the baby. And at that point, we say that those totipotent cells have become pluripotent, right? They have multiple, pluri just means potential into many, 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 but not all cells, right? Totipotent is all, pluripotent is most. You can think of it as most. From pluripotent cells, the cells are going to continue to differentiate, become different from each other, continue to specialize. And now we say that the cells are multipotent. So a multipotent cell would be a cell that can become different types of blood cells. Or a multipotent cell would be a cell that could become different types of connective tissues, but it can't become everything. Multipotent cells then further specialize to become oligopotent. Oligo means just a few. Oligo means just a few, or a limited number of cells. And then finally, most cells go on to become what we call unipotent. That means they only have the potential to become one kind of cell. So let's take a look at this continuation, and actually, let's take a look at this example. We'll come back and study this chart more completely when we get to the blood. But here we have one cell. This one cell is a multipotent stem cell. This cell is found in your bone marrow, and this one cell can become multiple different kinds of cells. How this is working, we're not going to worry about, but this one cell can receive different chemical and hormonal signals, and those chemical signals will change it, differentiate it into different cell types. And we see that this one blood stem cell can become all of the different types of blood cells in your body. Now, you haven't learned these cells, so I'm not even naming them. But all of the different types of blood cells in your body come from one cell. Now, what happens if one of these cells goes nuts? What if it loses control of its cell cycle? Well, then we have leukemia or lymphoma, right? These are cancers of the blood. And what happens is that one of these cells loses control, starts to make millions and millions and millions of itself. It basically crowds out all the normal blood cells. Normal blood cells can no longer be produced, and these whacked out, highly fast-growing precursor cells are the cancer. So, differentiation is simply the process by which a cell becomes more specialized. Think, I mentioned the other day that kidney cells and liver cells are different from each other. Well, how is that? Kidney cells and liver cells have exactly the same DNA in them. Remember, every cell in your body has exactly the same DNA in it. It has the same collection of genes. The difference is a kidney cell will only turn on some genes, and a liver cell will turn on different genes. That means that the kidney cell will make different proteins than the liver cell. And those differences are important in defining what a kidney cell does versus what a liver cell does versus what a skin cell does, or a stomach cell, or a brain cell, or a muscle cell. So cells become different from each other. They go through the specialization or differentiation process, and different genes are turned on. That's what this is referring to. Different genes are turned on or off to create those different types of cells. And here's just another picture of the same story. We start off with a totipotent cell. It has total potential. It then breaks into 3 different types of pluripotent stem cells. We'll use these words later in the next presentation. I'll use the words endo and meso and ectoderm. And those pluripotent cells then become more and more specialized into multipotent cells, which eventually become single kinds of cells. Isn't it neat to think that all of the cells in your body, right? You've got lung cells and pancreatic cells and heart muscle cells and red blood cells, skin cells, neurons, and many, many others. All of those different cell types all came from you when you were a single cell organism. So when your mom's egg and dad's sperm got together and made the zygote, right? Those cells, it divided 2 cells, 4 cells, 8 cells, 16 cells. And through this process, all the different kinds of cells in your body have been formed. So that's just a quick story through this idea of cellular differentiation. That finishes the textbook portion of this. But I know that this section of the text and this content is for many of you confusing. It's something you've all had, but for many students, it just doesn't stick. And the reason is you can't touch it, right? When we get into anatomy in about 2 weeks, we're going to be touching things. We're going to be touching bones and muscles, and we're going to be talking about brain and heart structures and parts of the stomach and parts of the kidneys and the liver. But for the next couple of weeks, right, we have all of this book knowledge, and a lot of students just struggle with this. So I'm going to jump over to some PowerPoint slides. These are posted for you in your folder. They're just underneath the video link for this presentation. And I'm going to go through these very quickly. There's some extra words here. There's some extra images here. Most of this I've already said, so I'm really just going to review this sort of a second time. So what is the cell cycle? The cell cycle is the time that a cell lives, right? Survives in your body. And cells live for only a finite amount of time. The cell will continue to grow, make new cells, and the organism will replace old and damaged cells. The way by which cells are replaced normally is through a programmed cell death process called apoptosis. So apoptosis is programmed cellular death. And here's a good example of apoptosis. We're looking at a human embryo from week 5 over to week 10. if you look at the early embryo, the arm and the legs are barely beginning to form. We call this the arm bud and the leg bud. And over the next few weeks, right, the arm bud elongates and you see the arm get longer and longer and longer. And by the time we finish up this period, from week 5 to week 10, this is a close-up picture of an embryonic hand at 10 weeks of human development. Here on the bottom is a embryonic foot, right? And you see the perfect picture of a foot with all of the toes perfectly formed. These embryonic feet are sandwiched between an adult finger and an adult thumb, right? Just to give you a sense of size. So what happened during this 5 weeks of development? Well, some cells grew and some cells died. We probably think that from the time we're an embryo until we're born, we just keep dividing and dividing and dividing and dividing. But no, that is not the case at all. Many cells undergo apoptosis. In the 5th week of human development, there are no individual fingers. We call this a paddle. And what happens is that over the next 5 weeks, some cells divide and make more of them. Other cells actually die. And because some cells died, it forms the separation between the fingers and between the toes. So our body is constantly undergoing a process of cell division and programmed cellular death, apoptosis. I think this is just a really cool example of apoptosis in your own developmental lifespan. Another time where we really care about the cell cycle, and we'll be describing the reproductive system at the end of the course. But here's where your blessed life began, right? This is representing the female reproductive system. Here is the uterus. Here's one fallopian tube. There's another one over here that we're not seeing. And at the end of the fallopian tube, we have the ovary. So here the egg was released from the ovary, picked up by the fallopian tube. Sperm have already arrived up the vagina, up the uterus, through the fallopian tube, and right up about here is where sperm always meet the egg. So fertilization in humans happens right here at the very top of the fallopian tube, what's called the ampulla. So the egg is released, the sperm fertilize it, looking at this picture here, and now that is called the zygote, right? The zygote is the first cell where the egg and sperm have come together. And then rapid cell division occurs. That zygote splits through mitosis to become 2 cells. Those 2 cells become 4 cells. Those 4 cells become 8. And we continue until we're a little ball of cells called the morula. Now, while the morula is being formed, the embryo is coming down the fallopian tube. It takes about 5 or 6 days for the embryo to come down the fallopian tube and reach the uterus. By the time the embryo reaches the uterus, it has already undergone one change. Remember I told you that some cells become the placenta and some cells go on to become the baby? Well, already by the first 5 or 6 days of human development, these blue cells represent the cells that will go on to become the baby. The rest of the cells, this capsule around, is going to go on to become the placenta. So these cells here in the morula are totipotent. The cells here in the early blastula or the blastocyst, these are pluripotent. Okay? And these cells are going to develop into you and me. That all happens here in the uterus. The embryo attaches, the placenta will be formed that will connect the baby to the mother's blood supply. So again, we really care about early development. If this cell cycle does not work properly, then obviously the embryo will not be formed well enough to implant onto the mother's uterus. And so we don't know exactly how many, but we know that oftentimes an egg is fertilized but it does not properly implant onto the uterus, and that embryo is simply lost even before the mother is aware that fertilization had occurred. Pregnancy is not the same as fertilization. Fertilization is when the egg and sperm come together. That happens probably a lot more often than we know. Implantation, when the embryo implants into the uterus, that is now the beginning of pregnancy. So again, I just bring this up so you appreciate, why do we care? Why do we care about learning about the cell cycle? Well, man, we all are dealing with the cell cycle every day. And in addition to the devastating news of cancer, we also can think about the joy and the wonder of early life as the embryo comes down the fallopian tube. So as I mentioned before, normal tissue has a normal balance of cell division and cell death, or apoptosis. But if that balance is upset and cells begin to multiply faster than they die, then we're going to get a mass of cells called a tumor, right? That's what a tumor is. It's one cell that went nuts, lost control of its cell cycle, and now is growing out of control. Now, cancers come in two basic flavors: benign and malignant. Benign. Benny, you're learning from your vocab, Benny means good, right? A benediction is a good word. A benign neoplasm, if you're going to get a cancer, you want a benign tumor. A benign tumor is still cancer. It's still a growth, but benign tumors typically are confined. They do not spread. They grow much more slowly. And usually they're not lethal. So if someone has a benign tumor, right, typically we can go and we can remove it. The only time that a benign tumor is life-threatening is if that benign tumor is deep in the brain or someplace where it cannot be safely removed without causing further damage. So in those cases, it's a very slow-growing tumor, and oftentimes they'll simply watch it, but typically benign tumors are not life-threatening. But if you hear that you have a malignant neoplasm, this is where you are very concerned. Mal, M-A-L, means bad, right? These are the bad news bears. These are the bad news cancers. And the deal with malignant neoplasms or malignant tumors is that they are unencapsulated. That means that the cells can spread much more easily. I had this on the previous slide. I didn't tell you about this, but cancer cells also tend to de-differentiate. In other words, cancer cells, rather than becoming more and more specialized, actually become more and more childlike. It's like they go backwards in their developmental pathway. And we all know that younger cells grow faster. So most cancer cells de-differentiate. In order for a tumor to survive, it needs a blood supply. So malignant tumors have a special capacity to increase their vascular supply, to increase their blood supply, right? Cells need oxygen, cells need nutrients, and tumor cells also need blood supply and oxygen. These cancers grow very quickly, and they tend to spread. And the word for the spreading of a malignant cancer is metastasis. So malignant tumors oftentimes metastasize. They spread to different places in the body. We've already said cancer cells lose control of their cell cycle. Here's a fact that I want you to appreciate. Most cells, normal cells, right, cells that are in your body right now that are normal, have what's called contact inhibition. When a cell bumps up against another cell, that's sort of a polite signal, right? Stop growing. So when a cell touches another cell, it stops growing. Think about your skin, right? You have all these cells in your skin, and when they touch each other, they stop growing. But cancer cells don't play by the rules. When cancer cells grow and they touch each other, they're not inhibited by touching, and they continue to grow. That's why they make these balls, these tumors, right? They grow over and out of control. Like I said, they revert to a de-differentiated state, to an earlier specialized state. They tend to produce chemicals that increase their blood supply. I've already used that term, but I didn't give you this word. Angiogenesis. Angio, you're learning from your vocab on the first exam, means blood vessel. And genesis, in the beginning, the formation of. So the formation of new blood vessels is angiogenesis. And cancers have a special knack for increasing angiogenesis. Like I said, these cancer cells oftentimes metastasize as they become invasive. They're able to travel throughout the body where normal cells would not go. So that's just a quick little recap on cancer and the cell cycle, and I hope now you understand more why we are learning about the cell cycle. Let me also recap a couple more features about the processes: replication, transcription, and translation, and why we care. So replication I mentioned in the previous presentation. Remember, replication is the process by which cells make a replica, an exact copy of the DNA. In bacteria, for example, E. coli, bacteria can undergo replication at about 500 letters per second. That's amazing, right? Can you imagine typing 500 letters per second? Now, humans, we're not that fast. In human cells, we can only copy about 50 letters per second, but man, that's still good, right? Can you imagine being able to type 50 characters per second, right? So that's fantastic. That means about the fastest that a human cell can divide is about 8 hours. Some of the most rapidly growing cells in your body are your skin cells, or the cells that are lining your tongue. Remember, we talked about if you burn yourself in your mouth, those cells replace themselves very, very quickly. The cells that line your intestines and your stomach also grow very, very quickly. It takes about 8 hours, right, for them to go through their cell cycle, so that but they have to replicate all the DNA before they can go through this. So 50 nucleotides per second. Amazing. And again, here's the cell cycle. And I'm just pointing out one thing that I said earlier, that this process of DNA replication happens right here in the S phase. So just what I'm adding here, that in the S phase, that's where DNA replication occurs. The problem with replication happening so quickly is that just like if we were typing a lot of things, we make mistakes, right? Who doesn't make mistakes when they're typing? And when the cell undergoes replication and copies all of those ACTs and Gs very, very quickly, mistakes are usually made. It's awfully good. In you and me, replication makes a mistake about 1 every 100,000 letters. But let's face it, guys, we have trillions of cells with billions of letters, and so every time our cells divide, they do make a few mistakes. Now, those mistakes you and I get to know as mutations, right? Mutations are simply, muta means change. A mutation is nothing more than a change in your DNA. Most of the changes in your DNA are simply the result of normal replication and the errors that happen. Now, if you have a mutation in a skin cell, you're not going to pass that mutation on to your kids, right? If you look on the back of your hand and you go outside and you get too much sun and you get a sunburn, and that sunburn causes a mutation in your skin cells, that skin cell may become prone to cancer in the future, but that mutation is not going to be passed on to your children. The only mutations that can be passed on to your children are the mutations that happen in your egg or sperm, that is in your germline. So somatic cell mutations are not heritable, right? You can't pass them on to your kids, but germline mutations, changes to egg and sperm, are heritable. Where do these mutations come from? Right? Most of the mutations that occur in our body are happening during replication. Other errors or other mutations come from environmental damage. Right now, you're breathing. Remember I talked about those reactive oxygen species? Just by breathing and doing our normal metabolism, we're creating molecules which are damaging to our DNA. So we're picking up those mutations. We're also sitting under the sun, right? We're getting x-rays. You go to the dentist or whatever, you get an x-ray. All of those chemicals, carcinogens, smoke, all of those things over time change our DNA and lead to an increase in mutations. But here's the good news. Although we have we have all these mutations happening all the time. Right now, right, every second, you're probably creating a new mutation in your body. But we can fix them. Our body has about 130 different enzymes that are able to fix and repair mutations. Now, occasionally, those mutations slip through the repair cracks, and we end up getting a mutation which leads to a tumor. But most of our damage, most of our mutations are fixed. Another place where we get mutations are from our parents, right? Let's face it, we inherit mutated genes. We inherit genes that are already making us predisposed to diseases. There are childhood forms of cancer. And those kids more than likely inherited a gene from their parents which made them more likely to get that childhood form of cancer. Most of us carry 5 to 10 disease-causing genes in our genome. The good news is cancer rates are rather low when we're young. They're not zero, but they're low. And as we get older, though, what happens? As we get older, the incidence of cancer goes up. And so we see esophageal, rectal, skin, prostate cancer. As we grow older, and as our cells have undergone more exposure to carcinogens and to x-rays and ultraviolet radiation, and as our cells have undergone more replication cycles, more and more mutations accumulate, and over time, that can lead to cancers. This is an amazing statistic. Every second, you have in your body about 25 million cells undergoing mitosis. You have 25 million cells that are dividing every second. Amazing. That also means you probably have 25 million cells that are undergoing apoptosis, right? They're undergoing programmed cell death, because if we kept making more cells, we would just become larger and larger and larger beings. So as all of these cells are undergoing mitosis and mutations are occurring, the cell has 3 choices. Number 1, the cell can repair the damage, right? We have mechanisms to repair the damage. Number 2, if the mutation is so significant that the cell is going to be significantly damaged, sometimes a cell will undergo its own apoptosis. It will actually sort of commit cellular suicide and undergo apoptosis if the mutation is significant. Now, if the cell doesn't repair it and the cell does not die, then that mutation becomes permanently incorporated into your DNA. And again, these mutations over time are the basis of genetic changes and disease and genetic mutations. Let me just recall that, so that's our conversation on mutations. Let's talk about this process of protein synthesis. Remember, it's 2 things. It's DNA undergoing transcription to mRNA, mRNA undergoing translation to make protein. Here's a recap of that, right? That transcription happens in the nucleus. Double-stranded DNA makes single-stranded mRNA. That mRNA squiggles through the nuclear pores, comes out, hooks up with a ribosome. The ribosome is now going to translate the mRNA sequence to make the protein. So DNA is a nucleic acid, mRNA is a nucleic acid, and proteins are made up of amino acids. They are a different kind of molecule altogether. So DNA to RNA, that is transcription. RNA to protein, that is translation. Completely changing the characteristics of the molecule. This is just a different picture. Here we have the DNA in the nucleus, transcription occurring, number 2. mRNA is made, comes through the nuclear pore, hooks up with a ribosome. Here's the tRNA that's going to come down and match up with the codon sequence on the mRNA, and it carries with it the correct amino acid. Those amino acids are linked together to make the growing protein molecule. So why do we care? 5 minutes and we're done. Why do we care? Here is the DNA sequence, right? Transcription occurs. We make the mRNA broken up into segments of 3 called triplets or codons. And then the mRNA is translated to amino acids. How does that happen? Okay, there's 3 RNAs involved. I've already mentioned them. There's a video in Blossom about these. mRNA, that's what's made by transcription on the DNA. Then there's the tRNA, it looks like a lunar landing molecule. And then there's the ribosomal RNA, which is an important component of ribosomes. I've already shown you that slide. Sorry about that. And how does the ribosome know what to do? It turns out that those 3-letter codes mean something. So every time the ribosome sees, for example, let's go here, AUG, it knows to put the amino acid MET there. Every time the cell sees ACU, it knows to put in threonine. So there's a genetic code, and this is the code right here. So the genetic code is the blueprint, the plan by which the cell knows which amino acid to add in. I'm going to skip over this slide and this slide. And I just want to go, and it's this slide as well, and I want to go to here. And let's figure out why we care about mutations. I think this will make sense now. So here is the normal mRNA sequence for a gene. We've already undergone transcription. This is the mRNA. This is the 4 amino acids that would be added in through the process of translation. What happens if there's a mutation? What happens if there's a change in the nucleotide sequence? Well, if you change the mRNA, that's going to change the amino acids that are built to make that protein. And so by taking away a letter, that's a mutation, you are going to get a garbage protein, right? And let's look at a couple more of these examples. If you remove or delete or change any of these mRNA nucleotides, then it changes the end result of the protein. If that protein is important for, let's say, the making of sugar or the making of a critical protein in your metabolism, then you would no longer have that protein and you would have a disease or maybe even have a condition that is not sustainable with life. So every time there is a change in the DNA, that DNA change can have an impact on the amino acids. And that's really why we went through this very basic story about transcription and translation. Here's a disease that I want you to just know the example of. And the disease is sickle cell anemia. Sickle cell anemia is a mutation, it's a genetic condition by changes in the protein called hemoglobin. Hemoglobin is the protein that carries oxygen in your red blood cells. And in normal hemoglobin, part of the DNA has CTT in it. And CTT on the mRNA would be GAA, and GAA will always be coded in as glue by the ribosome. In sickle cell anemia, instead of CTT, there's a 1-letter change. It changes to CAT. That changes the mRNA to GUA, and that changes the amino acid from glue to val. That simple change, that 1-letter change in one gene, causes the blood cell to behave differently and to change their shape. And these sickle cells clog up our capillaries. They cause tissue damage. They oftentimes cause a premature death in the individuals who have this disease. So here are normal red blood cells, and here are cells that are sickled, and you can see that they are much longer. And as a result, they clog up and get stuck in the blood capillaries, and a tissue that can't get oxygen and can't get blood is going to undergo tissue damage. So just another example of these changes in the DNA sequence that changes protein. Here is a quick slide review. So what I want you to do is just go through and ask yourself, what are each of these organelles? Okay? And as you go through this, these will be labeled for you. So I'm putting this here as a placeholder for you, and you'll be able to go through and practice these different organelles. So that's just a review. And then finally, I'm going to put this here, and then I'll review this again next time. But on the first exam, you're going to be asked to write 3 sentences using these terms. And I'll describe this again next time. But you're going to write 3 sentences that use 10 of these 13 terms correctly. And these are all terms that we've talked about in the last few days. Transcription, translation, DNA, mRNA, cytoplasm, genome, replication, right? So you're going to write these words into 3 sentences. Remind me, but I will also remind your SI leaders to introduce this assignment to you, but this will be a written portion on exam number 1. So again, these slides are posted for you at the bottom of, or right underneath this video posting. And I hope that all of this information is making sense for you. And I'll talk to you soon.